Good morning, everybody. Just camera a little bit. Today we're doing chapter 6. Good morning, Bella. How are you? Can you hear me okay, Bella? Okay, great. I'm guessing you don't have a microphone, so I'll keep the chat window open. Okay, we are doing chapter 6 today. Hope you have that ready to go. I'm going to go through this pretty quickly. So if you miss something from what I'm presenting, the recording will be available later today, and you can fill in anything that you might miss along the way. Okay. Do you have any questions before we get started, Bella? Okay, here we go. Chapter 6. Okay, this is what mitosis looks like. We're going to be looking at this under the microscope in lab together in class pretty soon. These are the different phases. Okay, so let's go through the notes. You carry out a chromosome. So chromosomes are made of a chemical called chromatin, which is a combination of DNA that we've already talked about, and protein molecules. Protein molecules are there to help organize it during cell division. They're not visible until in a cell until cell division. So here you can see them. They look like purple spaghetti. So I'll be referring to purple spaghetti a lot. You can't see them unless cell division has begun, and that's usually in prophase, which I'll be talking about here in a moment. Okay, this figure that you see here on the right will be a figure on the test. I'll be asking you to identify the different parts of it. So let's go through it. When 2 loops of DNA coil around 8 histones, this is called a nucleosome. Your notes may say 4 histones. Please change that to 8. So a nucleosome is basically one of these things here. I'll be putting the labels in towards the end. When the nucleosomes coil together, they form a tight helical fiber, which you can see right here. Again, I'll put the labels in a bit. When the tight helical fibers coil into larger coils, they're called supercoils, which is this. So what is the point? The point is taking this very long, thin material and coiling it around and coiling that around in such a way you can take an extremely long, thin thing and make it into an easily movable object when you're trying to move things around in the cell during cell division. The supercoils are what you see during prophase and make up the chromosomes. So these are the purple spaghetti. If you guys saw that earlier, what looks like purple spaghetti, that's this. And then they coil up into these chromosomes when it's actually time to separate them. Before a cell divides, it duplicates all of its chromosomes, resulting in 2 copies called sister chromatids. So let me put the labels in. There's your DNA double helix. This is DNA, greatly magnified. So these are histones, which are proteins that help the DNA coil up. When you coil DNA around 8 of these histones, it forms what is called a nucleosome. When these nucleosomes coil together, it forms a tight helical fiber. When you wrap these tight helical fibers together, it forms what's called a supercoil. And this works kind of like how rope works. If you ever look at rope, it's made up of very tiny threads, which are woven together, and then those weaves are woven together, et cetera, until you get a whole rope. Then when you take the supercoils and squeeze them together, you get what are called sister chromatids. And the way that you want to remember this is in the same way that identical twins are very exactly the same as each other, these sister chromatids are identical to each other. And the spot where they're connected together is called the centromere. That's the spot where these 2 chromatids, which are these 2 identical copies, are stapled together. Okay, sister chromatids. So there's 1 chromatid. Now, there's something I'm going to ask you guys to learn, and that is the difference between a single burger and a double burger. Now, it may seem obvious, right? But here's the question. When you have 1 patty and a top and bottom bun, what do you have? You have a hamburger, right? What if you have 2 patties and a top and a bottom bun? You don't have 2 hamburgers. You have a double burger. I hope you guys understand that. If you have been to In-N-Out or any place that sells double hamburgers. So let's take a look at the figure and I'll tell you what I'm talking about. This is a single chromosome. This is a double chromosome, one double chromosome, in the same way 2 patties makes up 1 double burger. Now you can take this double chromosome, this duplicated chromosome, and turn it into 2 separate chromosomes. So this is 1 single chromosome. This is a double or duplicated chromosome, 1, and these are 2 separate single chromosomes. So try to remember that. When the cell divides, the sister chromatids separate from each other. So these sisters, there's a sister and there's a sister, making up a single duplicated chromosome. They separate from each other. So we have chromosome duplication. So we have 1 becoming 2 identical copies. Each of these sister chromatids is identical in every way to its sister. So if I ask the question, how are they different? The answer is they're not. They're exactly the same, as if a perfect photocopy in every way was made of the original. But what's interesting is the original actually gets separated so that there's half of each of these has part of the original in it. So it's kind of different. So these are called sister chromatids because they're identical to each other and stapled at the centromere here. Daughter cells are the result of the cell division in terms of the overall number of cells. So each of these is a sister chromatid, but after cell division, the cell actually pinches into 2 separate cells. Those cells are called daughter cells. So don't get sisters and daughters confused. Sister chromatids are made of DNA. The daughter cells are the result at the very end when the cell is physically separated. I'll show you that. So these are chromosome distribution to the daughter cells. Each of the new daughter cells gets 1 chromosome. So you started out with 1, now you have 2. They're each identical to each other. And it was used, the original was used to make a perfect photocopy of itself and put that into the new cells. Now, what is the point of mitosis, by the way? The point of mitosis is growth. So here's a little background. When on day 1 of each one of our lives, dad's sperm, mom's egg came together and you formed what is called a zygote. We're going to be talking about meiosis next time. But after that moment, when we're a fertilized egg called a zygote, all the cells in our body that became us are a result of mitosis. So all the growth from that first cell, all the maintenance, the growth of our hair and our skin and the inside of our digestive tract, all of that came from mitosis. So that's growth and development and repair. Meiosis, the other cell division process we're going to be talking about later, is how we make babies, how we make sperm and egg. That's it. So we're focused right now on how did we grow from a fertilized egg and how do we maintain our bodies? How do we heal? How do we make new skin and new hair? Okay. All right. This is a figure directly from your text. This shows the cell cycle. Eukaryotic cells that divide undergo a sequence of events called the cell cycle. And you can see that there's gap 1, synthesis, and gap 2. This is all part of interphase. And then the mitotic phase is this little teeny bit right here. Mitosis, which are the other cells besides interphase, and then cytokinesis, which is the physical splitting of the cell. The cell cycle consists of 2 distinct phases. Interphase, which is made up of gap 1 right here. Synthesis, where the DNA is copied, and gap 2 here. The mitotic phase is made up of the stages prophase, metaphase, anaphase, and telophase. We're going to go into that, but you can see it right here, mitosis, the mitotic phase. Telophase includes cytokinesis, which is the physical splitting. Cytokinesis or cell splitting happens at the end of telophase. So this cytokinesis is the end of stage T or telophase. So this little way of remembering things, I'll pay more attention tomorrow. I get this all the time from students, but it's a way to remember the stages of mitosis in order. I'll pay more attention tomorrow, interphase, prophase, metaphase, anaphase, and telophase, the end of which is cytokinesis, or the cell going from 1 into 2 and separating. All right, interphase. The nucleus and nucleolus are visible. So the nucleus is the big circle, and the nucleolus is the little circle inside the nucleus. Sometimes incorrectly called the resting stage. Why is it called the resting stage? Because you can't see what's happening inside. Scientists know from doing experiments, but you can't see it under a microscope that anything significant is happening. What happens during this stage? DNA is replicated during the S stage. So that means all the DNA is copied because we're going to go from 1 cell to 2. Each cell needs a full set of DNA in it. All cellular organelles are replicated. For example, all mitochondria are doubled. All chloroplasts are doubled if it was a plant cell. the cell gets bigger because when it cuts in half, it can't be half as big as it needs to be. So it actually gets a little bit bigger before it splits in half. It prepares for division. So everything is getting ready for cell division to begin, the actual mitotic phase to begin. So what are some other things that you should know? Centrioles. These are special structures that work kind of like fishermen. So I'm going to give you this analogy of the fishermen, and then we're going to use that analogy to help us understand what they do. And these centrioles are going to help organize the separation of the copies. Remember, we have 2 copies of the DNA after synthesis. We've got to separate them apart so that each set goes into the new daughter cell, so that each one has its full set. So these centrioles work kind of like fishermen. I'll give you that analogy as we go. They're going to start to form the spindle apparatus, which you see are these little strings sticking out of it. I'll show you how that works. All right, prophase. So if you remember purple spaghetti, right now we have black spaghetti. It depends on how it's stained. So here we can now see that the DNA has coiled up into these supercoils. And when you stain it, you can see it under a microscope. The nuclear membrane starts to disappear. Why does that happen? Because you're going to have to pull these chromosomes apart. And if they're stuck inside of a bubble, you can't separate them. So the nuclear membrane begins to disappear. The spindle apparatus begins to form. So this is the structure that's going to pull the copies of DNA apart at the end of this process. So this is what it looks like under the microscope. We're actually going to see this in lab. And this is what it looks like in a diagram. So you can see the spindle apparatus starts to form. It looks about like the shape of a football. But what I want you to think about is each of these strings is going to work kind of like a fishing line. And as you see, these strings are starting to hook onto the chromosomes. Notice one string at one end is hooking onto one of these sisters, and the other string from the other side is hooking onto the other sister. So what I want you to imagine is you may have been to the beach and seen fishermen throwing several fishing lines out into the ocean. One of them contains several different lines. So if they catch a fish, they can go to whichever one has the fish on the line. So these fishermen at each end of the cell have all of these lines, each attached to a single sister chromatid. So notice that when it all gets lined up, this centriole is going to be hooked onto the top sister chromatids, and this centriole is going to hook to the bottom. How are they different? They're not. They're exact copies. So the idea is when you start to pull, this copy goes up, that copy goes down, and you separate them. You're going to see how this works later on. Okay, so the next stage is metaphase. Sister chromatids line up the equator, which is the center. The equator in our planet is right in the center on the outside. So it's the center part of the cell. The spindle fibers attached. Now you can see very clearly that all the spindle fibers from the top centriole are attached to the top sister chromatid, and the bottom centriole has attachment to all the bottom sisters. The centromere is the location where the 2 sister chromatids are still attached to each other. So right at the centromere is where these spindle fibers attach. So they're going to pull the copies apart. Now, notice that these images here actually under the microscope look a little bit different than they do in the diagram. That's because the tails are not attached. Only the centromeres are attached, and the ends kind of float around like they're in the breeze. So here it shows them perfectly lined up because it's a diagram. In reality, only the center of the centromere lines up, and the tails of these chromosomes kind of dangle out. So this is what it's going to look like under the microscope, and this is what it looks like in a diagram. Anaphase, the next stage. So anaphase is where the copies are being separated apart. In lab, I'm not going to have you look at early. We're going to be looking at late only, because this looks kind of like prophase. Now notice late, we have 2 bundles that are moving in opposite directions. So this is one full set of sisters, and this is another full set. If you look, you can see there's an equal number of the different lengths on both sides. So the spindle fibers contract. These little fishermen are winding their strings in. The sister chromatids separate at the centromeres and move towards the poles, which are the ends, kind of like our planet, North Pole and South Pole, equator in the middle. So notice that the diagram here looks a lot more similar to the actual microscope images, but you can see one sister chromatid is going up, the other sister is going down. So by the time they organize down here, there'll be a full set below and a full set above. Telophase. Now, if you look at telophase, you can see there's a little line forming between this clump of chromosomes and this clump of chromosomes This is what is called a cell plate, found only in plants. Chromosomes begin to clump back into a ball. Why do they do this? Because you're going to have to form the nucleus around it, and that protects the chromosomes during normal cellular activity. The nucleus begins to reform. The cytoplasm begins to split. This is called cytokinesis. So this is where the actual cell, instead of just being full of chromosomes at either end, physically splits into two cells. Cytokinesis is different in plant and animal cells, and I'll go into how it's different here in a second. Now notice the plant cell is building a little wall in here, and that's logical because the plant has a plant cell around it. If you guys remember an earlier lecture, they have cell walls. So the analogy is like if you wanted to divide a large room into two smaller rooms, you would just build a wall down the middle. And that's just what plant cells do. This is the cell plate, which is going to become the cell wall that separates these two new cells. But imagine if you wanted to separate a balloon into two functionally separate balloons. What would you do? You'd tie a string around it and knot it or twist the balloon. So what's happening here is this, because it doesn't have a cell wall, these little fibers are going to start to contract right in the middle, cutting this balloon, animal cell like us, into two. Okay, so in an animal cell, this is also a figure from your text. What forms is what is called a cleavage furrow. So the cleavage furrow is where it's going from one ball into two. So now from one, it's become two. So it's basically like tying a rope around a balloon and cinching it, where one balloon then becomes two. So you can see as this contractile ring goes, eventually it'll separate into two. In animals, the cleavage furrow forms, pinching the cell like a balloon. And in plants, as I said, because there's a rigid cell wall, these Golgi vesicles start forming the cell plate. The cell plate becomes the new cell wall, and now you have two new cells. In plant cells, the cell plate forms. In plant cytokinesis is more like dividing a room in your house by building a wall in the middle, as I told you about before. Okay, moving on, cancer. So cancer is related to mitosis because it's unregulated cell division. Now, normally, your cells wait until they get a signal to divide. Hey, we need more cells, and the cells say, okay. But if the cell loses its ability to stop dividing, it's like losing your ability to stop on a hill, because there's a special mechanism inside of our cells that allows it to stop between cell divisions and wait for further directions. In cancer, that stop sign signal inside of our bodies gets destroyed or disabled, so that when it goes at the end of one of the mitotic phases, it just loops right back into another one. So what is cancer? Cancer is caused by a breakdown in the control of the cell cycle. That is, the ability to stop and wait for the signal to divide again is disrupted. Cancer cells divide excessively, so they're going to continue to divide. Cancer cells can spread from a malignant tumor. And when that happens, metastasis means it's spread. Now, how does it spread so easily? Often through the lymph vessels. Now, the question is, why does cancer kill you? We know that people die from cancer, but why is that the case? Well, here's the deal, guys. Normally, all the cells in our body have a particular job that they need to do so that you can stay alive. All the cells in your body have a job to do. And if they don't do that job, they're not helping you stay alive. Now, cancer cells, instead of doing the job of the body, their only job is to divide and take resources from you. So imagine if all of your cells, or many, many, many of them, started to rebel, and instead of helping you, doing their job to keep you alive, their only job became to acquire resources and to divide as fast as they can. When that happens, not only are you losing resources from the other cells that are trying to do their work, but the cells that were supposed to be doing work aren't doing their work. So you lose a lot of resources, and the function of your non-cancerous cells become blocked as well. So when that happens, you can die from it, because too many of these cells stealing too many resources and not doing their necessary important job is too much, and it can kill you. Now, let's talk about how metastasis works. There's a tumor. Some of these cells are going to break loose from the tumor. Because you have a system in your body called your lymphatic system. You guys have heard of lymph nodes. Well, cells can go right into this and spread and get stuck in your lymph nodes because it's like a filter, and they can grow there. It's called lymphoma. If they don't get stuck there, they can get stuck in other places and start to grow. That's one of the main ways that cancer can spread. It's called metastasis. And the different stages talk about how serious and difficult it is to treat. So how do we treat cancer? Well, the evolution of cancer treatment is underway, but one of the traditional ways to do it is radiation. So if you hit cells with radiation, it will kill them, especially if they are dividing. So what they have done traditionally is bombarded either the entire body or more recently just the tumor with lots of radiation so that these rapidly dividing cells will die. The problem with that is that if you don't target just the tumor, the cells around it that are dividing will also die. Chemotherapy involves drugs that disrupt cell division. So basically the idea is the drug doesn't know whether it's a regular cell dividing for good reasons, looks like our hair and our skin and our digestive tract. Most chemotherapy drugs now will kill all cells that are dividing. Well, that's really hard on the body because we do need new cells on a regular basis. We need more blood cells. We need more skin cells. We need more hair cells. So think about the fact that people's hair falls out when they're getting chemo. That's because these drugs don't distinguish between normally dividing cells and cancer cells that are rapidly dividing. Now, what's even more ironic about this is that what are two things that can actually cause cancer? Radiation and toxic chemicals. So you're basically trying to fight fire with fire. You're putting radiation in, which can cause cancer, and you're putting toxic chemicals in, which can cause cancer. What the modern version of cancer treatment is called immunotherapy. That is getting your own immune system to find cancer cells and kill them, which we do pretty well when we're young and not so well as we age. So immunotherapy is the future of it. And if you guys remind me, I will put an extra credit question on the next test that has to do with this chapter about what is the state of immunotherapy cancer treatment. If you guys look it up and summarize it in your own words, you can get extra credit on the next test, but you may need to remind me about that. Cancer cells are often grown in culture for study. So they sometimes will take cancer cells, remove them from a person, continue to grow them so they can learn how cancer works and how to treat it. Okay. All right, so that is it for today's lecture. Do either of you have any questions for me about that or what's going on? I guess it is, when are we meeting? Monday for lab? So I really need everybody who's watching this to come on Monday. We are doing a lab. I did do a lab on Monday, but only had 2 people show up. So I need you guys to show up because I'm not going to be doing an alternative activity like I did this Monday again next Monday. So you guys need to show up. If you don't show up, you won't be able to do the lab. If you don't do the lab, you won't be able to turn in the lab report. You won't get the points. Your grade will drop. So please, everybody, come to lab on Monday. We are going to be having an actual lab that will require a lab report. Okay. Do either of you have any questions before we call it a day? Rihanna or Bella? Okay. Well, thank you both for joining me. Thank you, everybody, for watching this. And work on your review questions. Work on the Labster lab for this week. And I hope to see you on Monday. Have a great rest of your week. Great weekend. See you on Monday, guys.